Hello friends, now we are looking at the third part of our chapter sexual reproduction in flowering plants, and we've been talking about the different parts of it. We've been talking about the pre-fertilization event. We talked about the stamen and associated development and pistil and associated development. Now it's time to understand the idea of pollination and outbreeding devices. Now both of these terms are really important just to understand before the double fertilization event. Double fertilization is a specific type of fertilization event, or what we can say the fusion of male and female gamete in flowering plants. And I told you, like, the fertilization event takes place twice. That's why it's known as a double fertilization. But before the fertilization event, we all know what fertilization is. It's simply the fusion of male and female gametes together. So let's assume these simple terms. Let me draw the idea. This is the egg, this is the female part, and this is, let's say, the pollen grain. Okay, this is the male part. It's very important that pollen grain interacts with the egg, and actually pollen grain start to form this pollen tube, with which the nucleus from the pollen grain start migrating inside the egg and bind with the egg nucleus there. This is the overall event in double fertilization, which we will see in the next part. But the question is, before the fertilization event, it's very important that the pollen grain should come in contact with the egg. But what we know is that if you look at the structure of female flower, now what we can see is that this egg is present further downstream or further down region of this flower anatomy. Let me show you that picture as well. I just deleted that picture, it was there from earlier. This is how it looks like. There is a stigma, there is a style, and then there is this ovary. So this is the ovary, and this is the place where the egg is there. Okay, so this is where the egg is, and the pollen grain needs to in contact with stigma, and this one is style. So this pollen grain needs to be in contact with the stigma part of the female flower. Okay, or let's say stigma part of any flower which carry both male and female parts. Now this process is known as the pollination. Well, the pollen grain will be in contact with, this is the pollen grain, will be in contact with stigma. How exactly plants achieve that? There are different types of plant out there. There are plants who can have flowers containing both male as well as female anatomy. Both are present. They are known as hermaphrodites because they have the capability of producing the male gametes as well as capability of producing female gametes. So it's very easy to bring male and female gametes together and fertilize. And there are other types of plants who has either a male part or a female part in the flower. We call them unisexual plants or dioecious plants. Those dioecious plants, for example, papaya. So one type of papaya tree, they have only male part in the flower. Another type of papaya, they have only female part in the flower. So what they need to do? From one male flower from a papaya, the pollen grain needs to be transferred to the other papaya flower with the female part. And that is a hectic job, although very important job to be done. And who does that? There are two types of way this process of pollination is possible. So pollination is possible by two ways: abiotic factors, with the help of abiotic factors, and with the help of biotic factors. Abiotic factors, example, air, water. These are the two very common type of abiotic factors that may help to disperse the pollen grain so that the pollen grain becomes in contact with stigma. And biotic factor means insects, right? Insects that are linked with transferring pollen grain from the anther of a male flower to the stigma of a female part of that same flower or for a different flower. That what's happened in the pollination process. So in abiotic factor is very common because air is always circulating in, so it can take pollen grains, spread pollen grains outside. But depending upon which factor that the plant is using, the structure of the flower is different. If the flower utilize abiotic factors like air to transfer seeds, in that to transfer pollen grain, sorry, in that case they need to have the structures of all these pollen grains need to be attached very lightly, because whenever the microspore are dehydrated, they will make the pollen grain, and the pollen grain should become loosely attached to the rest of the part of the anther so that it can be dispersed by a little air or little flow of air. That's what the idea is. Now if a plant is using abiotic factor like water for transfer of all these pollen grains, in that case they also need to have a proper structure to the pollen grain so that the pollen grain should make the journey, or it can make the journey throughout the water, because in water there are different drawbacks and all the Brownian motion and all the other physical motions are as well present, so they need to survive that. That's why whenever we studied the pollen grain structure, I told you they have this two layer, and the outermost layer of the pollen grain is the most resisting material that we find in the entire world, right? That is the reason behind this, because they need to survive a lot of rough environmental situation, fluctuating temperatures, acidity, alkali and pH concentration, which is high pH or basic concentrations. All these things they need to survive to go until they can meet the egg. So that's why it's different. And for this type of plants who always utilize abiotic conditions, abiotic factors like air and water to transfer the pollen grain to the female part of the plant, they don't need to make attractive flowers, because they don't need any fragrance to generate. So they are mostly, sometimes they are not very good looking flowers, and they don't develop any fragrance as a flower that we know, right? Because they don't need it, because the idea is air will always be present, water will always be present. Doesn't matter whether the flower is colorful or produce a good fragrance or not. But on the other hand, trees who utilize biotic factors like insects to transfer the pollen grain needs to be colorful, and they need to produce fragrance, scent that helps to attract all the pollinators. Example of such different insects like butterflies, bees, and the other type of example is also beetles. Okay, so mainly butterflies and bees, they are playing a very important role in as pollinators, because they can grow from one flower to the other flower from different trees in a particular area. So when a bee is sitting on one particular flower and touching that flower's male part or anther, then what happens, let me draw the anther as well here. Let's say this is how the anther will look like. Let me draw it here, like, okay. So here these are the small what I can say, the pollen grains which are linked here, present here like this. So let's say a bee is sitting here. So now the anther, the pollen grains now attached with the leg of the bee on the complete body of the bee, and now the bee is transferred from there and sit on top of the stigma of the female flower of the same species plant. Then what will happen? That particular pollen grain will now be deposited on top of stigma. Now if that pollen grain is the right pollen grain to initiate the process of fertilization, then this female part will accept it, and first they will check the compatibility. It's very important that whenever a pollinator transfers the male gamete on the top of stigma, then the stigma has the unique capability to find out whether the pollen grain is of perfect nature or not, or compatible nature or not. Because if it's not compatible, then they will not allow this particular pollen grain to initiate double fertilization. If it's compatible with the mode of different cell signaling process, this plant female part of the flower, they find this out that yes, this is the compatible one, then it can start. It is allowed to create this pollen tube throughout, and finally can reach the egg, and the nucleus is transferred finally to fertilize the egg that is present in the ovary, inside the ovule obviously. So these are the steps of all process. And this is very, very important. People didn't know this idea regarding how exactly they determine whether the correct pollen grain is in place or not. Now they determine that with the help of different range of signaling. The exact process of that signaling is not well understood though, but we know that there is some sort of signaling that they check before inserting that pollen tube inside through the style, finally reaching the ovary. Now this is the whole idea of this transfer. But remember, when pollinator sits there, they can do this job. Now there are two ways that this job can be done. I told you earlier as well. One of the way is the abiotic way with air and water. No pollinators are required, no insects are required. So what are the difference between those two types? Abiotic and biotic. In abiotic type, this is not controlled by any other insects or any other third animals to do the job. So will it be beneficial for the plant or not? Now the answer to that, for many plants, they rely on abiotic factors because they can't produce that good quality fragrance producing flower. But many flowers, in fact most of the plants utilize to produce a very good quality flower with better fragrance to attract all the pollinators, because somehow plants from the plant's perspective, pollinators and the efficiency at which they can cause the pollination is much higher than the random airflow or water flow, because the pollinators are surrounding the complete area of some flowers or some trees in the neighboring place. So the genetic diversity is well kept utilizing the pollinators. That's one important thing. Now apart from this classification of biotic and abiotic pollination, there are also further differences regarding the pollination. So let me tell you about the classification of the pollination as well. The classification of pollination can be divided into three major sections. One is the autogamy. Okay, autogamy is a process where the pollination takes place in the same plant and even in the same flower. That's the autogamy. Autogamy occurs to only those plants who has the flower containing both male as well as female part. That means both with stamen and pistil, because in that case both of the structures are present in same flower. It's simply the transfer of the pollen grain from the male part to the female part, and that's the job done. That's how easy it is for the autogamy to occur. And this autogamy can be of two different types. For few cases of the plants who are utilizing autogamy, they have the open flower system. That means the open male as well as open female parts in the flower. That's how they are arranged. And there are few type of plants where they have complete closed system, because as they are following autogamy, they don't need to open up their petals and showcase the male and female reproductive part of the flower's structure. So they can completely close it down, because whatever thing will happen, it will happen in that same flower. So closed flowers are always possible. So there are these two types. One, this chasmogamous, another one is the cleistogamous. So chasmogamous and cleistogamous are two different types. Chasmogamous are the types where the petals and all the male and female parts of the flower are open, right? Are open and visible from outside. While the cleistogamous type of plants, in cleistogamous type of plants, they have complete closed system. So the plant, none of the male and female reproductive part of a flower is exposed. It's completely blocked. You can't see it from outside. So these are the two types of plants, but both follow autogamy because both has the male and female reproductive system in the same flower. This is one example. The second type is geitonogamy. Geitonogamy, let me write it down. Let me write it here. The first one, autogamy, geitonogamy, and another one, the last one, xenogamy. These are the three types. So you've seen autogamy and two examples of that. Now geitonogamy and xenogamy, these are the two types. In geitonogamy, the idea is also very similar to that of the autogamy. In geitonogamy, it's also self pollination. That means in geitonogamy as well, the pollination takes place in the same plant but with different flowers. In autogamy, the male and female structures are present in a same flower, so they are kind of bisexual in nature. Okay, okay. So for autogamy, for geitonogamy, flowers are separate. Male flowers are separate, female flowers are separate. Okay. Now pollen grain from male flower will transfer to the stigma of a female flower. That's geitonogamy, but in the same plant. Remember that. So autogamy, same plant, same flower. Geitonogamy, same plant, different flower. And xenogamy is known as a cross pollination. So this is cross pollination. These two are self pollination. Okay. Cross pollination means whenever pollen grain from one plant is transferred to another plant of the same species, obviously, because the reproduction will take place between the same species individuals. But let's say this is from plant A, so the pollen grains from anther from plant A is transferred to the top of the stigma of plant B from different plant. That's why it's known as a cross pollination. Okay. Now what is the difference between self pollination and cross pollination? Because in self pollination it's not that unique. Cross pollination is much better in terms of genetic variability, because self pollination, what happens is between the same plant, so the chance to generate variety is very less, because whatever structure or whatever chromosomes are present in the nucleus will remain as it is, because we are doing the cross between the same flower of a same plant, right? So it's not going to change. So it's going to produce genetically identical structures of the plants. But with xenogamy, there is a chance that a different feature from a different plant is transferred to the other plant. So the offspring may have more chance to be little different from the parents. That's why, because sexual reproduction is all about producing varieties, and these variations accumulated to cause evolution, and that will help the plant to grow better day by day. But which is not mostly true in case of autogamy and geitonogamy. It's true for the xenogamy. That's why cross pollination is mostly important. So if cross pollination is important, then why plants undergo autogamy and geitonogamy? Plants try to prevent the chance of self pollination. It never wants self pollination. To prevent self pollination, they have different techniques, different approach. We call them outbreeding devices. So outbreeding devices means different approaches plant takes, with which they can prevent they can prevent the self pollination, because they love to have cross pollination, because self pollination is possible, although cross pollination is much better in terms of genetic stability and importance. So these outbreeding devices, I am going to say 4 such different ways that a plant try to prevent the self pollination. Okay, one of the ways, for self pollination, both this male and female part of a flower should be present in same plant. So what they are going to do, because whenever there is a air or things like that, the anther can easily land on top of the stigma. So to prevent that, what they can do, there is a specific time duration for which the stigma remains active for the chance of fertilization, because this egg is not always active till eternity. There is a time duration till which the egg is ready to be fertilized, and if the time is passed, even though you put pollinators and even though you put all the pollen grains, fertilization will not take place. So what they do here, they change the time. It's a temporal separation there. So what happens, whenever the pollen grains are coming from the anther, at that time the egg lost its viability to be turned and to be converted and for the process of fertilization. So no fertilization is possible. Or similarly, it can also take place like if you change, like at the moment this egg remain viable, the anthers and the pollen grains have not matured enough, so they are not released. So the change in time can help them to minimize the self pollination. This is one way. The second thing that they can also achieve for outbreeding devices is also changing the mechanism of activity. For example, in many plants, whenever the pollen grain lands on stigma, that pollen grain somehow is not allowed to form the pollen tube through the style, okay, to prevent the self pollination. because they create a specific sense that if the pollen comes from the same plant, they are not going to facilitate the reaction to cause the entry of the nucleus inside the egg. So they prevent it unless a new pollen grain from another plant, from a separate plant, comes in to create a cross pollination. Okay, that's another way that they can do. Now the third important thing is, sometimes the biggest advantage that a plant can play with this idea of cross pollination is simply make their flowers only unisexual. That means a plant with only male flower, another plant with only female flower. So that's not going to create any problem, because if one plant have only male flower or only the male portion, then obviously they cannot self pollinate. So they need to transfer the pollen grain to another plant with the female flower in it. Then only the process can continue. That's why they produce these two separate type of plants known as staminate and pistillate. Staminate with only the male part of the flower, and pistillate which carry only the female part of the flower in that plant. That is also the best way to prevent the chance of self-pollination, and always try to achieve the cross-pollination. This is another very important idea. And the fourth way that they can prevent the self-pollination is by changing the position of how the male part and the female part of the flower are arranged. Even in the same flower, if they are arranged in different locations, for example, if anther is present little lower and the stamen and stigma is on top of it, on high regions, then what will happen? Try to assume this idea that this is the pollen, this is the anther, and this is the whole stamen, and this is the stigma placed on top. Then what will happen? If stamen is present on top, then it's very easy for the pollen grain to drop on surface of stigma. But if it's the differences in their height, it's not possible always. Another way is that they keep the stigma away from stamen. They keep the stigma away from the anther part of the flower, because if they keep it away, so that the pollen grains, even though there is air, but the pollen grain cannot take entry and cannot attach to the surface of the stigma. That's another way. It's a mechanical way. By changing the position of stigma and changing the position of anther, they can prevent the chance of self-pollination. And plants are really working hard to get rid of the self-pollination, because cross-pollination is more important. Cross-pollination can change the genetic makeup. It develops new genetic variations which are required, and which is the actual motto of sexual reproduction, which is the actual idea behind sexual reproduction in plants. So these are the 4 outbreeding devices that we talked about. One is the change in the morphology and the position of male and female portion and parts. Change in timing for the process of double fertilization, and also changing the behavior or the chemical pathway with which the pollen grain can be recognized by the female part. And the fourth is changing the type of flowers that they have. So unisexual plants like staminate and pistillate. Okay, so these are the 4 ways outbreeding devices are performed, and those all help a plant to be more cross pollination, to use more cross pollination and less self pollination. Okay, so that's our idea about pollination and outbreeding devices. If you like the video, please hit the like button. So in the next part of the tutorial, we'll be talking about the double fertilization with an animation, and then we'll also talk about after the fertilization what happens to that whole embryo, what happens and how the embryo develops, what happens to the endosperm that's created, what happens to the embryo, how the seed of a fruit is formed, and how and what fruit is really about. We'll talk about all these things after the next lecture. Stay tuned. Flower plants undergo an unusual reproductive process in which there are 2 fertilization events rather just one. This double fertilization takes place between cells produced in the male reproductive organ, the anthers, and the female reproductive organ, the ovule. The ovule contains a reproductive cell called the megaspore mother cell or megasporocyte. This cell is diploid and undergoes meiosis to produce 4 haploid megaspores. In most species, 3 of these megaspores degenerate, leaving only one surviving megaspore. The one surviving megaspore then undergoes 3 rounds of mitosis to produce 8 haploid nuclei. Cytoplasmic divisions do not follow immediately after the nuclear divisions, so the 8 nuclei initially share the same cytoplasm. The multinucleate structure is called embryo sac. Within the embryo sac, cell walls form between most of the nuclei. 3 cells, known as antipodal cells, form opposite the micropyle opening of the ovule. 3 also form near the micropyle. 2 are called synergids and the other is known as the egg. Finally, 2 nuclei called the polar nuclei remain together in one large central cell. The egg cell and the single cell with 2 polar nuclei will eventually take part in a double fertilization event. Before this can happen, the male gametes, the sperm, must travel to these cells within the female's reproductive organ. To reach the embryo sac, a pollen grain that has loaded on the stigma begins to germinate, sending a long pollen tube through the style and the ovary. A haploid cell called a generative cell travels down the pollen tube. The generative cell travels behind the tube nucleus, which is the nucleus of the large cell that makes up the bulk of the pollen tube and grain. The generative cell divides by mitosis to produce 2 haploid sperm cells. The pollen tube reaches the micropyle of the ovule and digests its way into one of the synergids, releasing the sperm cells. The synergid degenerates as one of the 2 sperm cells fertilize the egg cell, producing a diploid zygote with 2n number of chromosomes. The second sperm cell fuses with both of the polar nuclei, fertilizing them to produce an unusual triploid 3n number chromosome containing cell. As the zygote develops into an embryo, the triploid cell develops into endosperm, which serves as embryo's food supply during the growth.